The work that I've done on the disease has been based on artificial inoculations. As Jorge will tell you later on, he's seen no evidence for any difference among avocado varieties and their ability to attract and be poured into by the vector. So all the work that I've done, I've not used the insect. I've used only the pathogen to look for disease resistance and look at the efficacy of fungicides and that sort of thing. In all these experiments then, we've used a subjective rating scale. We first used a 1 to 5 scale, where 5 was dead and 1 was healthy. More recently, we've used a 1 to 10 scale. So on some of these graphs that I'll show you here after this slide, don't be confused, but it's either a maximum of 5 or 10, and 1 is always healthy in these areas. One of the first things we saw in our experiments and field studies is that there was a very profound relationship between plant size and severity of disease that develops after inoculation. They already knew this with red bay. They detected in the natural environment that large red bay trees developed this disease before small red bay trees, and they developed the disease much more severely than small red bay trees. We studied this experimentally with avocado and found the same thing, and the reason I'm mentioning this is that this makes it really difficult for me to do these studies, in that these large grafted avocados that are useful in disease studies are really expensive, and for some of the varieties that I'd like to study, they're just simply not available. So it's put a wrench in the works when I've tried to look at cultivar variation, variety variation, and how it responds to this disease. That said, we've looked at about 24 different varieties so far, and there are a couple things I want to show you on this. I don't want you to look at all these, but in the left-hand column, I've got the different cultivars, and then in the next column to the right is genome. That's just simply the relative contribution of these different races of avocado to the different cultivars. So for the dark green area down here, all these WIs are West Indians. West Indian race of avocado for you guys here in California is not that important. It's a little bit cold sensitive, and for some reasons you don't really use it here. For us, it's the most important set of cultivars in South Florida. It and the Guatemalan West Indian hybrids. Fortunately for us, Simmons down here, which is our most important, most widely planted commercial avocado, has been routinely shown to be highly susceptible to this disease. It gets hammered. And as a group, the West Indian group here, you see this mean down here, they are significantly more susceptible than the Guatemalan Mexican and the Guatemalan testing. Haas up here, the mainstay of your trade here, seems to be less susceptible than some of the West Indian and West Indian Guatemalan stuff we tested. So these studies are still ongoing. We still don't have good results where I can go to our growers and say, okay, cultivar X, Y, and Z we recommended over Simmons. We just don't have the information. And in fact, what these data show is that there's not good immune resistance in avocado to this disease, unfortunately. We're going to continue though looking, and one of these things that this table shows is that it suggests that there might be greater tolerance to this disease in the Mexican and Guatemalan race than in the West Indian race. So with that in mind, we're going to be selecting with a bias towards those in the future to see if we can identify anything that's useful resistance for the future work. I'm going to use this slide to introduce some of the fungicide work that we're going to talk about here. This, we have shown there's a very significant relationship between internal and external disease development. External disease would be the wilting and defoliation you see. Internal would be dark off and see that vascular discoloration. What we found is that although those two types of disease rating are significantly related, you get significant internal damage before you get any indication of external damage. So external damage down here, one would be completely healthy. You can see here that we actually get moderate disease develop internally before you see any external disease. This suggests to me, and they've seen this before with Dutch elm disease, is that it's really highly suggestive that once you see disease develop externally, using an effective fungicide or something else to manage the disease is probably not going to work because you've already got widespread and significant disease inside the tree. So let's keep that in mind. Done a lot of work with fungicides. And Jorge is going to tell you that he's done a lot of work with insecticides. We've tried to come up with a silver bullet that would be very effective way of managing this disease. And this has been frustrating work. This is just a summary of all the different chemistries that we've looked at. And the nutshell here is that triazoles and thiabendazole have good in vitro activity against this fungus. There are some other fungicides, for instance the strobilurids, that also are highly effective or highly fungitoxic in vitro. Put the fungicide in a medium and then let the fungus grow and then effectively. We found though, bring this up too, the major caveat here is that the infection cord, where the fungus is and what the tissue that we have to protect is in the xylem. So here's the xylem. Just think about this is a cut open tree. And then to the outside of the xylem, we have the vascular cambium, and then the phloem, and then the bark. So if we're going to be treating trees with these fungicides, we have to get effective doses way inside the tree, way in here. And that's been really difficult. The most efficient way, effective way to do that is with something called macroinfusion. Macroinfusion just means you're infusing or injecting large volumes of fungicide. You guys in the audience who have problems with phytophthora root rot, I know you've injected phosphonates before, very effective against phytophthora root rot. But you're injecting in relatively minor volumes of that fungicide in the trees. With these fungicides, you have to get really large volumes, gallons of fungicide in the tree. To do that is really a major problem. So there's a company up in Minnesota, Rainbow Tree Care. They devise this technique for doing so. Real quickly, schematically, what they do is they excavate around a tree and try to identify every single flare root that comes out of the base of the tree. Drill holes in each one of those flare roots, tap into these little T's, and then pressurize this entire system. These reservoirs of fungicide. And then infuse or inject into those trees through those nozzles in the flare roots, these fungicides. They know that on elm trees, oak trees, and sycamore trees up in the Midwest, they can effectively manage a lot of different diseases and some insect problems by using this technique. Our problem is that this is very time consuming. The first time I showed this slide to our avocado growers, they immediately said, we're not interested in this. This is going to be too expensive. We're not going to be able to economically use macroinfusion for injecting fungicides. Jonathan and I will talk a little bit later, Jonathan will mention too, the jury's still not out on this. We're hopeful that if we can identify a fungicide that has long residual activity in these trees and can come up with a way to rapidly treat trees, that still might be a viable option. Because the other options we have of getting large, efficacious amounts of fungicide in the xylem are really minimal. These soil drenches, we just pot studies, we have drenched onto the soil, these different fungicides, protect small potted trees by doing that. The more effective way of treating these trees is with the top of the bark application. We do something called petrobon. It's a surfactant that some of you may have used for treating trees with phosphonates. It's a way to get materials past that bark very in our hands, proconazole, thiabendazole, and some other triazoles.